Hey guys, it's Sonic again. I'm here with the latest installment of Science with Sonic. And this is the screencast of what we did in class today, Tuesday, January 31st. And the reason we're doing this one is to go over some of the basics of mitosis, otherwise known as the cell cycle or cell division. Now, if you take a look at this, there are 6 basic stages to the entire cell cycle, beginning with interphase and ending with cytokinesis. These 4 middle stages, if you will, pro, meta, ana, telophase, are actually what is considered mitosis, and you can see it's listed right here at the top. Sometimes scientists just refer to cell division as mitosis, and they may even include the bookend phases of inter and cytokinesis. But just so that you understand, technically, these 4 middle stages are what are defined as mitosis. But if we look at cell division in its entirety, this is asexual reproduction. This is just typical cells in your body that are reproducing. You need new skin cells, new liver tissue cells. They reproduce this way, where one cell simply becomes 2. Remember that this one or this one are not copies of the original. I'm sorry, let me restate that. These 2 daughter cells, neither of them is this cell, and the second is a copy. It doesn't work that way. This cell right here became 2 cells that are identical to the original one. The simple explanation for this, and we're going to go to our little interactive cells alive, and we'll try to make this really quick, and I'm going to show this animation. This shows you how going through the different stages cause one cell to become 2 cells. If we go back to the beginning, a couple key points to be made. First of all, you have these 2 pairs of structures in the center called centrioles. And these centrioles, if I actually give you a better picture of them, look like this. These centrioles are these little discs with spindle fibers that are extending out from them, sometimes called microtubules. And these spindle fibers and centrioles, notice the spindle fibers are kind of drawn over here. What you'll notice them doing throughout this animation is they'll start to migrate to either side of the cell. So as we step forward, you'll see them start to go. So keep in mind that the centrioles are doing that. They are heading to what will become 2 new poles either side of an existing cell, because that's where 2 new cells will form. But if we go back to the beginning here, you have a nucleus, and within this nucleus is a whole bunch of DNA. And all of that DNA is uncoiled. It is what we refer to as chromatin. And actually, now that I think about it, if I go here, I did a little picture that I drew in class that I can show you. It's this one right here, I believe. Yeah. I drew a little picture on the bottom, and I drew it with 2 different colors. And the idea being that during the interphase stage of the cell cycle, you've got all of this uncoiled sort of spaghetti mess of chromatin. And chromatin is your DNA. And during this stage, DNA double helix molecules are replicating. And they replicate by doing this. And I don't need to get too into it. We don't need to get too complicated. But this double helix right here unzips. And these amino acids and these nucleotides are splitting off into 2 separate strands, and those 2 separate strands find existing components within the cell to match up and form 2 new strands of DNA that are identical to this original one. Feel free to pause, rewind, and listen to that again, but to keep it super, super simple, the idea, and if I can go back to my picture, where did I put my picture? To put it, oh, there it is. To put it simply, the DNA in here has copied itself. And there are now 2 strands, a blue one and an orange one. And they are copies of the original. And what happens during prophase, which is this next stage, you get condensed chromatin. And those condensed chromatin strands form structures called chromatids. And each of those chromatids are sister chromatids to one another. They are identical copies. They're like twins. And here I show them, these chromatids, paired together and held together by a structure called a centromere, and collectively they are called a chromosome. So if we go back to our diagram here, interphase is the beginning stage. It is the longest stage of the cell cycle. It's hours and hours long, depending on what kind of cell it is. It could be 12 hours. It could be 15 hours. DNA is replicating, copying itself. When that stage is done, we enter prophase. Prophase is defined as the stage in which the nucleus is still there. DNA has condensed into sister chromatids. You can see them there. Centrioles are migrating to either sides of the cell. Spindle fibers are stretching out. And then what you get is this in-between stage. Sometimes you'll see it called prometaphase. The nucleus is sort of dissolving, for lack of a better term. It's reconstituting itself into other things, and it'll form again later. So it's kind of dismembering or dismantling. That's the word I was looking for. It's dismantling into component parts so that there won't be a nucleus temporarily, for a few hours maybe, depending on the cell. And you pass that stage, prometaphase, the nucleus is gone now. And notice that the chromosomes are beginning to kind of separate from even one another, and they're going somewhere. The spindle fibers are stretching out. The centrioles are on either side, because what's happening is you're getting towards this stage called metaphase, where each of the chromosome pairs are lined up. And that's a key stage. That is this middle part, where it's always defined as these little X's that go across the center. The centrioles are on either side. And these microtubules or spindle fibers create a path for those chromosomes to split. So if we continue, they split. And now they begin to migrate to either side. Now remember, each of them is a copy of one another. If you go back to that beginning stage back here, they were copying as just chromatin. And then they formed chromosomes that were copies of one another. And they lined up, and then they split. And each of those individual chromatids are now migrating to either side. And we call it anaphase when you get sort of that distinct impression that there are two sides. You know, they're more to the centriole than they are towards the center. And you can really see, oh, I can see it now. It's looking like it's going to become two cells. And sure enough, you get to the point where we hit telophase. Now, telophase is typically defined as you get this little impression on this animation, but the point is that the nucleus is beginning to reconstitute. It's forming again. And now it's forming on either side. And because it's forming on either side, you get two new nuclei forming two new cells. And you start to see what they call a cell plate, or a division between the cells. And then once it pinches off, right there, we now have cytokinesis, the official point at which, boom, we have two new cells. So essentially what you have is one cell became two. And neither is the original one. They're both copies of this one. And then, of course, these two cells become 4. 4 become 8, become 16, 32, 64, 128. And the whole idea is that, I can make this, can I make this bigger? Yeah, there I am. Each of those cells. And so, you know, we talked a little bit about meiosis, which is sexual reproduction. And when a sperm cell fertilizes an egg cell, then you have one cell, and that one cell becomes two, and that's meiosis, which we don't get really into, but just so you understand there's a difference. So those two cells become 4, become 8, become 16, 32, 64, 128, just like these cells do. And the point is, but in meiosis, that one single cell became 17 trillion cells, which is really crazy to think about. But again, back to where we were here, this is asexual reproduction. This is how cells divide. Okay. Now, I'm pretty sure I covered everything that I wanted to cover. We've got our DNA, our centrioles. Yeah, I think that's it. Oh, you know what? There's one thing I could go over. This right here. Notice I did show the answers here in class, but if you take a look at this, this sort of illustrates a very simplistic way where you've got, during prophase, you've got all your pairs, but the pairs are drawn in different colors to indicate that they are sister chromatids, copies of one another. Then during metaphase, they line up, and then you have anaphase where they split, and then here we've jumped to cytokinesis where now you have two distinct sizes, but they're drawn in two different colors to illustrate the fact that you have two new cells that are both copies, though, of the original cell. All right? As always, rewatch, pause, come back to me with any questions that you might have. All right? So take it easy.